Dear student, today I am going to discuss regarding your the chapters which deals with chemical structure and function of the DNA and RNA, then replication of the DNA, then transcription and translation, and then regulation of the gene expressions. So it is a vast chapters. Okay, so it's not possible to complete it. Therefore, today I will discuss regarding the DNA structure, function, as well as the replications. The rest portions I will take in my next class. Okay. So first, before going to the this structure of the DNA, let us discuss a little bit history of these nucleic acids. Okay. The two types of the nucleic acids are found. Two types of the nucleic acids are found. In cases of the cells of the all living organism, they are deoxyribonucleic acids and ribonucleic acids. Now, the nucleic acid was first discovered by Friedrich Schmidt in 1869 from the nucleus of the push cells and was named as nucleins. Kossels and Newman in 1894 identified four types of the bases in the DNA, namely adenine A, thymine T, guanine G, and cytosine C. Hammerstein in 1900 showed that DNA contains pentose sugars. Levine in 1929 demonstrated the pentose sugar is 2-deoxyribose. Next, Avery, MacLeod and McCarty in 1944 discovered that DNA is the genetic or the hereditary materials of all living organisms. Chargaff in 1950 demonstrated that the consistency to the base ratio of the DNA, that is A always pairs to the T and G always pairs to the C, and the base ratio, that is AT pair divided by GC pair, may vary in the DNA of different groups of the animals but is constant for a particular species. Franklin's in 1952 produced X-ray diffraction picture that indicates that DNA is a double helix. Watson and Crick 1953 deduced the famous double helical model of DNA. Now let us discuss the chemical compositions. Now, chemically, DNA is composed of three different types of compounds. So everybody know that one. Then first is the sugar molecule, and second is the nitrogenous bases, and third is your phosphoric acid. So first, let us discuss the sugar molecule. Now, sugar molecule represented by a pentose sugar, it is also known as 2-deoxyribose. Now, pentose sugar means a sugar which is consists of 5 carbon atoms. So in my picture, I have shown you carbon 1, 2, 3, 4, 5. Okay. So out of that 5 carbon in a 2-des position, there is a OH is replaced by hydrogen in case of the deoxyribose sugar. But it remains same as it is in case of the ribose sugar, that is 2 position will be OH. So depends on the deoxyribose and ribose I have given. Okay. So this is the first molecules which is essential for the DNA, that is the deoxyribose sugar. Second is the nitrogenous bases. Now nitrogenous bases. So nitrogenous bases are actually two types of the nitrogenous bases. Actually, we know ATGC, but in true sense, nitrogenous bases it is two types, known the purines and the pyrimidines. Now pyrimidines are single ring compounds. The ring is hexagonal and heterocyclic. It is composed of 4 carbon atoms and 2 nitrogen atoms. The atoms in the ring are numbered in clockwise. Nitrogen atoms are placed 1 and 3 position, and rest positions are occupied with the carbon atoms. Okay. So nitrogens are placed in the 1 and 3 positions. Mark it. Okay. And rest positions are what is occupied by the carbon. So pyrimidines are cytosine and thymine. Basically, in this is the DNA. The pyrimidines are cytosine and thymine. But in case of the RNA, thymine is replaced by uracil, the cytosine and uracils. Now, practically, we know the cytosine, thymine and uracil, but what is the chemical configuration of the cytosine, thymine and uracil? Chemically, all the three are what pyrimidine, but still there are some differences. What are the differences? Cytosine is 2-oxo-4-aminopyrimidine. Thymine is 2,4-dioxy-5-methylpyrimidine, and uracil 2,4-dioxypyrimidines. So the structures in my next slide, just I have given the structures. So here you will note the structures. Very nicely it has been given the structures, that purines and pyrimidine both are given. Let us first discuss the pyrimidine. Pyrimidine is a single ring nitrogenous compound, 6-member single rings. Further, a pyrimidine has 2 nitrogen atoms at 1 and 3 days positions. Okay. So basically that is cytosine and thymines. Okay. So one position, say this is the one position. So this is the one position and this is the 3 positions. So one position and 3 position that is occupied by the that is nitrogen. Okay. And rest positions are the rest positions are occupied by, already I have mentioned, amino group or oxo group, like that. Okay. So accordingly I have given the names. And purines are what? Double ring compounds, double ring nitrogen compounds. Each molecule consists of the 2 rings, one pyrimidine ring of 6 atoms and an imidazole ring of 5 atoms. The 2 atoms are numbered anti-clockwise in case of the pyrimidine ring, but clockwise in case of the imidazole rings. The 2 rings share the carbon atoms present in the 4 and 5 position. The nitrogen atoms in the purine rings are 4 in number, present at 1, 3, 7 and 9 positions. Okay. So adenine chemically it is known as 6-aminopurine, and guanine is chemically known as 2-amino-6-oxopurine. The structure is very nicely given here. Okay, in my slides. In my next slides, just I have given the differences between the pyrimidine and purine, the chemical structure of the pyrimidine and purine, and how to count the pyrimidines and the purine rings. That is also I have given. Okay, so all the structure it is given a laboratory R stage. Okay, not the D stage. So next components of the DNA is known as the phosphoric acid. This structure, the phosphoric acid, which is popularly known as H3PO4. Okay. Now it remains attached to the either 5' or 3' position of the deoxyribose sugar. Now this phosphoric acid, in my right hand side I have given a structure that this phosphoric acid, it either remain attached to the what 3' and next is 5'. Okay, the position of the phosphoric acid either 3' and 5' of the that is deoxyribose sugar. So popularly this link is popular in the phosphodiester linkage. And phosphoric acid structure just I have shown here. It consists of H3, the formula is H3PO4, and one phosphate is just the 1 oxygen and 3 OH groups. Okay. Next, another popular term that is known as nucleosides and nucleotides. So nucleoside means when a deoxyribose sugar is attached to a nitrogenous base. A nitrogenous base and a molecule of the pentose sugar is known as deoxyribonucleoside or simply nucleosides. Okay. The nitrogenous base attached to the one carbon atom C1 of the deoxyribose sugar through N glycosidic bond. So this that is a nitrogen base is attached to the sugar by means of the bond which is popularly known as N glycosidic bonds. In all, there are 4 nucleosides. Since there are 4 types of the nitrogenous bases, so naturally there will be 4 types of nucleosides in the DNA molecule. They are deoxyadenosine, adenine plus deoxyribose sugar, deoxyguanosine, guanine plus deoxyribose sugar, deoxycytidine, cytosine plus deoxyribose sugar, deoxythymidine, thymine and plus deoxyribose sugar. So they are popularly known as the nucleosides. Now what is the nucleotides? On the basis of the 4 bases, a molecule of the pentose sugar and the phosphoric acid form the nucleotides. Now nucleoside means when nitrogenous bases are attached to the deoxyribose sugar, whereas in case of the nucleotides, when there is nitrogenous bases are attached to deoxyribose sugar as well as the phosphoric acid, then it is on the nucleotides. So naturally there will be 4 types of the nucleotides, namely deoxyadenilic acid or DMP, adenine, deoxyribose sugar and phosphoric acids. Deoxyguanilic acid or DGMP, guanine plus deoxyribose sugar plus phosphoric acids. Deoxycytidinic acid or DCMP, cytosine plus deoxyribose sugar plus phosphoric acid. Deoxythymidinic acid or DTMP, thymine, deoxyribose sugar and phosphoric acid. So differences is very clearly it has been shown here between the nucleoside and the nucleotides. Okay, and remember one thing, that always these nitrogenous bases it attached to one position of the carbon atoms by N glycosidic bonds. Now here the chemical structure of the nucleotides, the purine that is ATP and GTP, and pyrimidine that is TTP and CTP, and in UTP also. Okay, so this is the chemical structure of the nucleotides. So naturally now we know now nucleosides and nucleotides. Now naturally it arises another important term. What is a polynucleotide chains? So polynucleotide chain. Let us discuss the polynucleotide chain. Now DNA is a macromolecule formed by linking of the several thousands of the nucleotides. They are called monomers or building blocks of the DNA. In a nucleotide, the phosphate molecule attached to the 5' carbon atoms of the deoxyribose sugar molecules or the 3' carbon atom molecule. The phosphate molecule of the nucleotide joined by the 3' carbon atom and deoxyribose the next nucleotides with the 5'. Such 5' 3' linkage between the adjacent nucleotides are known as the phosphodiester bonds. Okay, because one phosphoric acid molecule joins to the hydroxyl group of the sugar molecule by ester linkage. The joining of the two nucleotides forms a dinucleotide. Of the three nucleotides, a trinucleotide, and so on. Short chains of the formed of less than 20 nucleotides are known as the oligonucleotides, and chain longer than 20 nucleotides refer to the polynucleotides. The one point it must be clear: one is phosphodiester bond, and one is simply that is known as phosphodiester linkage, and there is simply the ester bonds. Okay, so simply ester bond means when particular that means nitrogenous base. Okay, sugars, no, nitrogen, yes, sugar and phosphate is known as ester bond, but one sugar, when the two phosphate is in the phosphodiester bonds. But one phosphate is connected to the two sugar, then it is known the phosphodiester bonds. Okay, one is the ester, one is the phosphodiesters. So in my next that is slides, just I have given the different the structures of the polynucleotide chains. Now you see, in this structure is very nicely given the polynucleotide chains. Now one chain that is having the adenine, the complementary chain it will be a thymine, and say this is the starting chain, and the last ending chain, say guanine, it will be attached to the cytosine. So A always pairs the T and G always pairs with the C. In between AT there is a double hydrogen bond, and in between the GC there is a triple hydrogen bonds. Okay, and one strand if starts with the 5' it will end in the 3', and the complementary strand it will start with 3' and 5'. Okay, the reason obviously it is known to us because in the one strand starts with the 5' to 3', the complementary strand will be 3' from this. Why? Because the sugar, the position of the sugar, it will rotate such that the one positions will connect with the your that is nitrogenous base, and the three positions will be connected by your that is the phosphate bond. So naturally, when one phosphate in the one strand is a 5' position, and the complementary strand it will be at 3' position. That is what is popularly known as 5' 3' and 3' 5'. Now in my next slide, just I would like to give the important features of Watson and Crick double helical model of the DNA. Now according to Watson and Crick model, double helix consists of the two polynucleotide chains. Diameter is about 20 angstroms. The two strands of the double helix are antiparallel, that is one strand aligned in the 5' 3' directions, and the complementary strand will be 3' to 5' directions. Each polynucleotide chain has a sugar phosphate backbones. The phosphate group gives the molecular a negative charge. The nitrogenous bases is almost perpendicular to the long axis of the molecules, which forms the steps and joined by the hydrogen bonds. The steps are made of the AT and GC pairs. Each turn of the helix is 34 angstrom. The space between the adjacent base pairs is 3.4 angstrom. Therefore, 10 base pairs are found in a per turn. That is in 34 angstroms. Okay. The double helix of the DNA have two types of the groups. One is known as major groups, and there is known as major or the deep groups, and is the wide, and the minor groups which is shallow and the narrow. Okay. So in the next structure it will be clear what is the minor groups and the major groups. Okay. Now some more points. So DNA strands have a directionality, and the different ends of a single strand are called 3' end and the 5' end. The base sequence of the single strand DNA is left end of the sequence is 5' end, while the right hand sequence is 3' end. The strand of the double helix are antiparallel, with one begins the 5' to 3' and the opposite is 3' to 5'. Okay. Now the pairing of the complementary bases of the DNA through hydrogen bonding means that the information contained within each strand is complementary. The nucleotides on the single strand can be used as a reconstruct the nucleotides on the newly synthesized partner strand. There are alternate major groups and minor groups arise due to the helical structure of the DNA. Now, what is a double helix? What is a double helix? A double helix looks like a twisted ladder or a spiral staircase. Twisted ladder means jokhola, moi, or spiral staircase, shiri. Okay. So, double helix looks like a twisted ladder or a spiral staircase. The double helix accounted for many of the features in the Franklin's X-ray pattern and did not explain what forces held two strands together. That is, Franklin's could not define. Franklin's only discovered the X-ray, that is the double strand is there. But Watson-Crick found the answer. Watson discovered that the hydrogen bonds could form between the certain nitrogenous bases and provide just enough force to hold the two strands together. In between the AT there is a double hydrogen bond, and in between the GC there is a triple hydrogen bond, which explains by Chargaff's rules. Okay. So in my next slide, just I have given the ladder-like shape of the your DNA and the twisted structure of the also DNA. So in this structure, it is very nicely showing the one strand if starts the 5 dash, it will end in the 3 dash, and a complementary strand it will start the 3 dash and it will end in the 5 dash. And in a turn of the DNA, there is a one major groove and another there is a minor groove. The major groove is very wide and deeply it is situated, whereas the minor grooves it is not so wide and it is very shallow. Okay. So the diameter of the two strand is about 20 angstroms or 2 nanometer is almost the same things. And in between the one turn of the DNA, there is a 10 nitrogenous bases are there, and the differences between the two steps is 3.4 nanometer or angstroms. Okay, 3.4 angstroms. So in my next slide also I have given the helical structure of the DNA. So very nicely it will be shown the major groups, minor groups, base pairs, sugar phosphate, backbone, steps, all these things. Okay. Now this DNA always we discover that is always dealing with, that is the normal DNA, and this normal DNA always it is found in the B form of the DNA. So there are different forms of the DNA. Already you know that one. That one is B form of the DNA, and the A form DNA, and the Z form of the DNA. B form is the normal form of the DNA, where the one turn is about it measures that is 34 angstroms or 3.4 angstroms. But in case of the A form and Z form, it varies. Now the different properties of that is B form, A form, and Z forms very nicely I have given tabulated here. That will B form DNA, A form DNA, and Z form DNA. Helical sense, this is right-handed, this is right-handed, this is left-handed. Major groups in B form is present, A form is present, but in case of the Z form, which is absent, Z form is also on the RL helix. So base pairs is 10, it is almost 10 or 10.5, okay, 10, it is 11, it is 12. So glycylation bond conformation is NT, this is NT, this is NT in case of the pyrimidine, and SYN in case of the purines. Helix rise is 3.4 angstrom, this is 2.6 angstrom, this is 3.7 angstrom. So here naturally more steps are there, that is the number of the base pairs is also more 11s. Here is also number of the base pairs is more 12. Okay. Now this base tilt is 6 degrees centigrade. In case of the B form the DNA, it is 2 degrees centigrade and it is 7 degrees centigrade. The structure, this is the B form structure, this is the A form structure, this is the RL helix. Okay. So after that, I would like to discuss regarding the another important properties of the DNA, which is popularly known as the denaturation and renaturations. So we know that the DNA, as and when artificially it is heated, in vitro medium it is heated, it denatures, that means it breaks. Again, when we cool it, then it again renatures. So that property is known as the denaturation or renaturation, or it is also known as that is breaking of the DNA, okay, and that is annealing of the DNA. So the ordered state of the DNA helix, which is originally present in the nature, is known as the native form of the DNA. The two strands of the DNA readily come apart when the hydrogen bonds between these paired bases are disrupted. This can be accomplished by heating the solution of the DNA or by adding the acid or alkali to ionize its bases. The unwinding of the DNA double helix is called the melting. This is also known as the melting of the DNA. And transition from the native to the denatured state is known as the denaturations. Denaturation of the DNA molecule can be studied by measuring the absorbance at a wavelength at 216 nanometers. When a solution is there, that solution, it can be measured by 260 nanometer in the wavelength in a colorimeter or the spectrophotometer. The number of that means the more denaturation will be there, so naturally there will be more DNA. So that can be measured by a colorimeter or spectrophotometer of 260 nanometers. As the DNA is subjected to the increase in the temperature, a 260 absorbance 260 starts increasing because of the DNA. When both the strands are completely separated at a particular high temperature, there is a maximum absorbance 260 that indicates the complete denaturation of the molecule has taken place. The temperature at which the half of the helical structure of the DNA molecule is lost is called its melting temperatures. A convenient parameter analyzes the melting transition. Molecular rich GC pairs have the higher melting temperatures, and those having the abundance of the AT pairs because GC pairs are more stable and held together by 3 hydrogen bonds. Such DNA molecule require more energy and hence temperature to denatures. So what is the renaturations? So with the help of the diagram also I am showing the denaturation and renaturation. Denaturation means denatured DNA will renature. Okay. So when the DNA is broken down, again if you cool it, then the DNA it will again renature. That is popular on the renaturation. Or to reform the duplex structure, if the denaturing condition is removed, that is the solution cooled, the pH is returned to the neutrality or the denatured are diluted out. Renaturation requires reassociation of the DNA strand into the double helix, a process termed on the re-annulings. And for this to occur, strands must realign themselves so that the complementary bases are once again registered. That is the nucleation process. A helix can be zipped out. So this is the again zipped up. Okay. So don't use this figure 12.9. Don't use this one. Okay. Renaturation is dependent on DNA concentration and times. Many of the realignment are imperfect, and thus the strands must dissociate again to allow to proper pairing to be formed. The process occurs more quickly in the temperature is warm enough to promote the diffusion of the large DNA molecule, but not so warm, so as to cause the meltings. So in my next slide, just I have given the diagram. So DNA is the master molecule. Everybody will know this is the master molecule. A DNA molecule, it may synthesize the another DNA molecule, which is popular in the duplication or replication of the DNA. Which that property only it will be having in case of the DNA molecule, not in the RNA, nor protein, nor carbohydrate. Moreover, this DNA, one DNA it will give rise to the RNA, that is known the transcription, and RNA it will give rise to the protein, which is not translation. So in your syllabus, this replication, then transcription, then this translation, everything is there. So I will discuss one by one. So this is the DNA molecule. First there is a transcription. As a result of transcription, there is the formation of RNA. Okay. So there is the pRNA. Then this pRNA it will give rise to the mRNA. Okay, which will be capped and 5' is adenylate. This mRNA now come in the cytoplasm, and with the it will form the complex with the ribosome, and it starts the DNA synthesis, that is polynucleotide synthesis. So all this mechanism I will discuss one by one, chapter wise, and this is popularly known as central dogma of the molecular biology. Now according to the modern that is central dogma, there is simple differences there, that DNA give rise to the RNA sometimes, RNA also it may give rise to the DNA in case of the some viruses. So in my next diagram, so I am with the most important properties of the DNA, which is the DNA replication. So the DNA replication I will declare. Now one thing you try to understand, these DNA replication always take place in the synthetic period. In our cell cycle, this S period is there. That S period of synthetic period DNA replication takes place. DNA replication means from one original DNA, there will be synthesis of the two DNA, and these DNA replication is semi-conservative. That is, one strand will be parental strand, and the other strand will be newly that is synthesized strand, daughter strands. Okay, daughter strand will be new strand, and one parental strand. So from one DNA we will get the one DNA, that is two stranded DNA. We will get the new daughter DNA, which having the two molecules of the DNA. But in that daughter DNA, one strand will be always that is parental strand, and the two new will be the that is new strands. So that is the basic structure of the DNA replication, and that has been that is why it is known as DNA replication always takes place in a semi-conservative way. And Michelson et al. discovered these semi-conservative replications. Okay, now I'm going to now discuss all these things. I'm going to discuss mainly the how the DNA replication takes place. Okay, now DNA replication is the process of producing the two identical copies from one original DNA molecule. This biological process of replication occurs in all living organisms and is the basis of the biological inheritance. DNA is made up of the two strands, and each strand of the original DNA molecule serves as a template for the production of the complementary strands, a process referred to as semi-conservative replication. Most prominently, DNA polymerase synthesizes the new DNA by adding the complementary nucleotide to the template strands. In a cell, DNA replication begins the specific location or origin of replication. Unwinding of the DNA at origin and synthesis of the new strands results in the replication fork, formation of the replication fork, growing bidirectional from one origins. Okay, a number of proteins are associated with the replication fork, which helps in terms of the initiation and continuation of the DNA synthesis. Cellular proofreading and error checking mechanism also there ensure perfect fidelity for DNA replications. Everything is there, okay, in these master molecules. So it is continuing. So DNA replication can also be performed in vitro, that is artificially outside the cells. Okay, DNA polymerase isolated from the cells and artificial DNA primers can be used to initiate the DNA synthesis at known sequences in template DNA molecule. The polymerase chain reaction PCR is a common laboratory technique that is clinically or cyclically applies such artificial synthesis to amplify the specific target DNA fragment from a pool of the DNA. Now, during these DNA replications, the important mechanism, the DNA, before going to different mechanism, the most important thing is the what different kind of the enzymes. Okay, so different of the enzymes are what it is essential for this DNA replication or DNA duplications. Okay, now some of the important enzymes, I noted some of the important enzymes because there are so many different types of the enzyme. The first enzyme just I have mentioned here, topoisomerase, or that is known as DNA guidase. Now this topoisomerase or DNA guidase, it is a kind of the enzymes. It relieves the build of the torque during the unwinding. It relaxes the supercoiling of the DNA. Now DNA we know it is a very supercoiled DNA. Okay. Now where the replication will start, replication bubble starts. Okay. The topoisomerase bind two sides of this that is a replication fork. Okay. So that replications bubble will form, and this guidance will minimize the what? Minimize the it minimizes the relaxation of the supercoiling of the DNA. Okay. Next, helicase or DNA unwinding protein. At least 9 proteins are associated with the DNA helicase enzymes. This protein recognizes the origin of the replication and help in the unwinding, that is breaking of the hydrogen bonds, and the two strands will be separated. Okay. Now single strand binding. Once the two strands are separated, now each strand will be what? It will be stabilized by the single strand binding protein, which is popular in the SSBP. Okay, single strand stabilizing binding protein, so that the DNA replication it can carry on, or one strand it acts as a template strands. It should be a template strand, should be very stable one. Next enzyme is the primase, or it is popularly also known DNA dependent RNA polymerase. They synthesize the RNA primer for the attachment of the DNA nucleotide. So another important thing that when the DNA replication starts, it needs a primer. Okay, and what type of primer? This is the RNA primer. The very peculiar thing, this type of the RNA primer formations, or this is the primer formation, is not found in the other molecules. It's only found in the DNA molecules, RNA primer. And RNA primer synthesis takes place with the help of the DNA dependent RNA polymerase enzymes. Next is DNA polymerase 3. It is also called the replicases. It is the main DNA synthesis enzyme which elongates along with the primer with the DNA nucleotides. Okay, DNA polymerase 3. Analyzing the DNA polymerase 2, it helps the mismatching repair in the gaps of the DNA replicases. If there is a mismatching is there in the DNA polymerases, DNA replication, that the mismatching is repaired by this DNA polymerase 2. And there is another enzyme which is pulled on the DNA polymerase 1. It performs the cleaning function such as the proofreading. It also degrades the RNA primer. This is on the DNA polymerase 1. And lastly, you will get the DNA ligase or DNA annealing proteins. Okay, it forms the gaps between the Okazaki fragments in In the lagging strands, we know that synthesis is not continuous, it is discontinuous. Naturally, there is so many gaps are there. That gaps are what? It is that gaps are ligated. Okay, that Okazaki fragments are ligated by this DNA ligase. So here I have given some of the most important enzymes of DNA replication. It's not necessarily that this is sufficient. In addition to that, you can add more. And here I already have mentioned that the helicase of DNA anion putting at least 9 protein or enzymes, 9 types of the enzymes are there. So I have not given details so that it will be very complex. So I have given the main enzymes which is essential for the DNA replications. So in my next diagrams, I have given the DNA replications. Okay, how the DNA replication takes place. Now we see in this green one, which is the topoisomerase enzyme. When topoisomerase enzyme binds, the DNA is uncoiled DNA will it will be that is when topoisomerase binds, it will relieve the DNA so that less strains falls on the other part of the DNA. DNA is a long molecule, so first is binding in the topoisomerase. So after binding in the topoisomerase in the leading strands, there will be binding in the DNA polymerase, and then helicase, then single strand binding proteins. Okay. And naturally DNA primase, then RNA primase synthesis starts. Okay, then DNA ligase, it will come to act. Okay, DNA polymerase. So all these structures just given in this diagram. In my next diagrams, just I have given there is two structure of the DNA. Now it is shown in the first structure. Just I have shown this is the unwounded DNA. This is the unwinding DNA. This unwinding DNA. First you see topoisomerase is the protein that binds to the double helix head of the replication fork and relieves the strain placed on the double helix. It is unravels. Okay, so this is the topoisomerase. Okay, so the arrow is there, so very nicely it has been shown. Okay, so topoisomerase binds two sides of the DNA so that less strain falls on the other part of the DNA molecule because it's a very supercoiled DNA. So next enzyme is known as DNA helicase, most important enzyme, DNA helicase, and breaks the hydrogen bonds. In between these two helix, there is a hydrogen bonds are there. So this hydrogen bonds, DNA helix breaks the hydrogen bonds in between the two strands. This allows the DNA to unwind into two strands at the replication fork. So this is the replication fork or the replication bubbles, and in the replication bubbles, that means I have shown the DNA helicases are acting, as a result of which hydrogen bonds are broken down. So down this replication fork or replication bubbles, it is very clearly seen in my next slides. The single strand, that is a binding protein, now it will bind to prevent the separate strand from the joining together again by the binding to separate strands and stabilize them. And once you stabilize it, then starts your synthesis of what new strands. So this is this now each strand will act as a template strands. Okay, template, on this template the daughter strand will be synthesized. Okay, in my next slide also I have shown that this is the different type of the phosphate, 1, 2, 3 phosphate. This phosphate is attached to the that is deoxyribose sugars at 5 position, and base is at the 1 position, so it will form the deoxyribose sugar. So naturally there is a 4 types of the DNTPs are there, which is essential during the DNA replication, but DNA synthesis. Okay, 4 types of the types are essential. Now in my next diagram, I have shown you see first there is a synthesis of this. This is the RNA primer. This is red one, 5' to 3'. There is a RNA primer synthesis. Okay, once the RNA primer synthesis is over, then start DNA synthesis. Now in replication, the 3' regions. This is the 3' regions. Okay, 3' regions. So 3' regions group of the last nucleotide on the strands attacks the 5' phosphate group of the incoming DNTPs. Okay. Now 2 phosphates are cleaved off and 1 phosphate will be added. Okay, in the DNA. As a result, the deoxyribonucleotide triphosphates, that is from this deoxyribonucleotide triphosphate DNTP, only the one that is a phosphate will be added, and the 2 phosphate will be released because it will perform the function of the energy. Okay. So next is what a phosphodiester. In this way what will happen, in the fourth diagram, a phosphodiester bond forms between the two nucleotides. Okay, so and the phosphate ions are also released. So here this is the what diagram of mechanism of the replication. This is the schematic models. So in my next slide also I have given another that is structure of the what model of the that is the DNA this replication. Now in the first position, on the upper template strand, the DNA synthesis proceed continuous in the 5' to 3' direction, same as that of the unwindings. Okay, so this is the template strand, this is also template strand, and this is the new strand, green color. This brown color is also new strand of the daughter strand. The direction of the DNA synthesis is always 5' to 3'. So if the direction is 5' to 3', the DNA will be parental strand will be always 3' to 5'. Okay. So in my next diagram, the second diagram, on the lower template strand, the DNA synthesis begins at the fork and proceeds in the direction opposite that of the unwinding, so it also runs in the templates. Okay, so naturally upper one is known as the leading strands and lower one is the lagging strand. In case of the lagging strand, DNA synthesis starts again on the lower strand, there is the lagging strand, been a small segment which is known as the Okazaki fragments. Okay, and this Okazaki fragment later it will be what ligated by means of that is ligating enzymes, that is DNA ligase. So leading strand and this is the lagging strands. So this is the leading strand and this brown color is the lagging strand. DNA synthesis on the lagging strand is discontinuous short fragments of DNA proceed by the discontinuous synthesis are called the Okazaki fragment. According to it is discovered by the one scientist in the Japanese Okazaki, and it is popular known the Okazaki fragments. Okay. So in my next slides, just I would like to summarize the mechanism of the replications. Okay. The process of by means of which the duplication of the DNA takes place is known as the replication. Replication is a semi-conservative and discontinuous process. During the process of the replication, the following events takes place. Okay. Now, topoisomerase of the guidance binds to the DNA strands and separating the two strands so that the DNA remain in relaxed configuration. Already I have shown in my earlier, that is a schematic view. Replication process is initiated at a particular point of the DNA known as the origin, which targeted by the initiator protein or single strand binding protein at the origin of replication or the orisites. Once the origin has been located, this initiated protein that the DNA helicase or DNA replicase unzips, uncoil. Okay, just like unzips means uncoil the double-stranded DNA or parental strand. As a result, Y-shaped fork formation takes place. In case of the eukaryotes, the replication starts at the multiple point in the nucleus, but in prokaryotes it is one or two sites only in the cytoplasms. Once the template strand is ready, the beginning of the RNA primer, the binding of the RNA primer takes place in the presence of the DNA-directed RNA polymerase into the 5' or 3' directions. So the template strand is 3' to 5' direction already I have shown earlier. After binding the RNA primer, DNA polymerase initiate the new daughter DNA synthesis. So synthesis of the leading strand of the RNA primer takes place 3' OH in the direction 5' to 3' in place of the DNA polymerase 3 by adding the nucleotides triphosphatase one by one to the growing template DNA regions complementary to the template strand. If there is A, then there is a T, then T, A, G, C, C, G in that way. Elongation of the DNA chain takes place by adding the nucleotides at 3' OH ends, so the leading strand synthesis takes place at 5' to 3' direction. Once the strand synthesis is over, RNA primer removed by 5' end by the action of the 5' series explicitly active with the DNA polymerase 1. It also acts as the proofreading in the elongation strand. Any mismatching in the DNA replication is corrected by the or is edited with the DNA polymerase 2. Okay. So next strands is known the lagging strands. So lagging strands. So lagging strand means once the that is first template strand is over. Now with the second template strand synthesis starts in the second template strand, but in the second template strand the synthesis of the DNA is not continuous, but instead it is discontinuous, and that is why popularly it is known the lagging strands. Okay, so lagging strand synthesis. What is the lagging strand? Once the leading strands synthesis is over, the lagging strand synthesis starts in a short separated segment 5' to 3' direction. On the lagging strand template, a primase that is the reads the template DNA and initiates the synthesis of the short RNA primer. A DNA polymerase extends to the prime segments from the Okazaki fragments. The RNA primer are then removed and replaced by the DNA polymerase, and the fragments of the DNA oncogenic fragment are joined together by a DNA ligase, which is not present in case of the leading strands. Now, DNA polymerase 3 in prokaryotes or Pol delta or Pol eta in eukaryotes is responsible for the extension of the prime segments. Primer removal of the eukaryotes is also performed by the Pol delta. In prokaryotes, the primer removal is performed by the DNA polymerase 1, which reads the fragments, remove the RNA using by the endonuclease domains. RNA primers are removed 5' to 3' exonuclease activity by the polymerase 1 and replace the RNA primer with the DNA nucleotides. So this is done the lagging strand synthesis. Okay. So this synthesis, that is DNA leading strand and the DNA lagging strand synthesis, popularly is known as that is the DNA replications. Okay, very in nutshell, just I have given what do we mean by the DNA replications. Okay, now there are some differences between the prokaryotic and eukaryotic replications. There are many origins of the replication in case of the eukaryotic DNA. So number of the replication forks are several, whereas in case of the prokaryotic DNA replication forks is 1 or 2. Replication bubbles are more in case of the eukaryotes in comparison to the prokaryotes. In case of the eukaryotes, instead the nucleus replication takes place, but in prokaryotes it is a cytoplasm, and since there is no nucleus. In eukaryotes, each replication consists of 50,000 to 3 lakhs nucleotide pairs, whereas in case of the prokaryotes it is 150 base pairs only. Okay. Origin recognition complex ORC are more in eukaryotes, very complex in the eukaryotes, is less in prokaryotes. DNA replication of prokaryotes is circular in the expression of circular DNA, whereas in case of the it is eukaryotes, it is known the linear DNA. So here